Tēnā koe, Professor Perry. Kia ora, koutou katoa, no mai haere mai. Welcome everyone. It's really great to see so many people here from within the university and also guests from outside of the university. This inaugural professorial lecture marks Nigel Perry's due recognition as a University of Otago research professor. And I'm going to come back to that classification a little later in my talk. A very special welcome to Professor Perry's wife, Tracy, and their children, Gabriella and Robbie. And where are Gabrielle and Robbie? Yes. Oh, fantastic. Thank you. IPLs are events that I personally regard as very special and the real highlight of the week. And the reason I say that is it gives me the opportunity to see some of our brilliant people who work at the University of Otago. And the other thing that it reminds me of is that being a university employee is not just about going to meetings, not just about convening boards of study, and it's not just about administrative problem solving, which is what I do a lot of at the moment. And it reminds me just how important good teaching and good research is to the University of Otago in terms of its reputation internationally. So inaugural professorial lectures are very special events, and tonight is no exception. And what we're going to do is, just in a moment, Professor Barker will provide a brief biographical background. And so I'm not going to focus on Nigel's stellar career. However, I will mention that he's rather unusual. And he's unusual because we share him with Plant and Food Research, which is a Crown Research Institute. And it provides research and development targeted on fruit, on vegetable and crop and food products. Nigel's a research leader in plant and food, but he's based in our chemistry department, where his research could never be really described as obscure and irrelevant because it supports the sustainable production of high-quality produce, driving the design and development of novel, functional foods that benefit human health and well-being. Specifically, Nigel researches natural products chemistry, which is all about the compounds that give colour and flavour to our foods, scent to flowers, and inspiration for many of our life-saving drugs. Now, because I chair the University Staffing Advisory Committee, I often talk about how difficult it is to be made a university professor. Yet, because teaching is not formally part of the research professor's job, I need to single that out as something slightly different. And to be a research professor, promotion is even harder. And it's harder because you have to achieve sustained performance at an outstanding level and also leadership in just two areas. And there's no slack. So whereas for a normal professor, you can get 2 out of 3, and the third one is not quite as high in terms of leadership. For a research professor, you only have 2 options, and there's no ability for the committee to move things around. So research professors are very special within the university. With nearly 150 refereed journal article publications when he applied for the position, plus applied spin-offs, Professor Perry clearly ticks the box for research. But his service as the head of Peru, which you'll hear about later, I have no doubt, including commercialisation, patents, contract R&D, plus postgraduate supervision, which incidentally research professors don't have to do according to their job description, but Nigel unreservedly commits to that. All of that is absolutely outstanding. And so when considered for research professor, it was a very, very easy decision. 4 referees supported the application, and we had glowing statements such as, "I give my greatest level of support for this most deserved promotion to the rank of professor." And Perry shows superior evidence of being beneficial and scholarly outcomes. In my opinion, he's already demonstrating the level of competence and leadership associated with a professorial appointment. Remembering, of course, that when he applied, he was not a professor. So what the referee was saying was that he'd already achieved that even before he applied. So I'm not going to say any more. I'm going to pass over now to the Pro-Vice Chancellor of the Division of Sciences, Professor Richard Barker, and he's going to tell us a little bit more about Professor Perry's meritorious career. Thank you.
Tēnā koe, Professor Perry. Tēnā koutou katoa. It's my great pleasure tonight to introduce our speaker, Professor Nigel Perry, to give his inaugural lecture at the University of Otago. Born in England, Professor Perry attended high school in Guildford, Surrey, before heading on to university where he studied chemistry. After graduating with first-class honours from the University of Bristol, Professor Perry began his career as a flavour chemist for Spillers Limited in the UK. And my little bit of research indicated that Spillers produced dog food or pet food, amongst other things. Motivated partly by the outstanding alpine climbing opportunities presented in New Zealand, Professor Perry wisely chose the University of Otago as a place to study his PhD. Graduating in 1984, he then commenced a lengthy postdoctoral fellowship at the University of Canterbury, which he commenced in 1984 and finished in 1990. In 1991, Professor Perry was appointed research leader at what is now Plant and Food Research, a position that he holds today. Fortunately for us, he only holds that for 80% of his time. The remaining 20% of his time he holds a research professorship in the chemistry department here at Otago. Professor Perry's research interests combine his understanding of fundamental chemistry with a strong drive to find practical applications. This work includes medical and agricultural research, and he's worked with a number of groups representing Māori interests here in New Zealand, as well as a number of international and New Zealand companies. He is currently engaged on research with Tito Ihu on Manuka bitters and Naitahu on perfume development from Taramea. Taramea is also known as wild Spaniard, and Naitahu have been working on this for more than 200 years. Professor Perry also works with Pipfruit New Zealand on sensing disease in apple trees and Phytomed Limited on relaxants from Manuka. Besides a significant body of published research, Professor Perry holds 6 patents, including one on an insect attractant now in commercial use around the world. For his research on the discovery and development of biologically active natural products, Professor Perry was awarded the 2013 Prize for Industrial and Applied Chemistry by the New Zealand Institute of Chemistry, an institute that has also recognised him as a fellow. He was awarded a New Zealand Science and Technology Medal for Excellence in 1984 in recognition of his work. This evening, Professor Perry will describe his journey from the University of Bristol to the University of Otago. Along the way, we're going to hear about examples that arose from his PhD research, from marine chemistry while he was engaged on his postdoc, and significant examples from his current research today: trees, sponges and bees. So please welcome Professor Nigel Perry.
Tēnā koutou. Thank you very much, Vernon. Thank you, Richard, for those introductory words. And thanks to you all for coming along. And thanks to the University of Otago for the great honour of appointing me to this research professor position. So I'm going to be taking this up, and thank you for the opportunity to speak to you tonight to tell you what enthuses me about natural products chemistry and to try and convince you that you should be totally interested in it. In fact, you are, without realizing it, because we all like food, beverages. Most of us like flowers, and that's what I'm going to be telling you, some of that. I'm going to run through briefly, as Richard said, 3 stories from my career. Working on trees, working on products from the seas, and working on bees, but I won't be able to rhyme for the rest of the lecture. And those stages take me from the University of Otago here on my PhD, and I'm going to explain to you, most of you will have seen this strange-looking sculpture, which is in fact a molecular structure on the end of our fantastic new Science One building that's been totally refurbished. And I'm going to talk about an antiviral natural product from our own Otago harbour out whichever way it is. It's hard to tell in these lecture theatres, isn't it? Then I'm going to talk to you about bees and the hidden danger that may be lurking in the favourite food for a lot of us, in honey. And I'm going to talk about unique New Zealand molecules, ones that some of them have still only been found in New Zealand. So all of them were first found here in New Zealand. And the techniques that we use in chemistry to discover these invisible molecules and work on them on a tiny scale. So why natural products chemistry? What is it? As we've said, it's a study of, one aspect is a study of natural colours, scents and flavours around us. And that, as Richard has said, that's why plant and food is one of our interests in that. We breed apples, we breed kiwi fruit, new varieties of wine, so we want to control and improve the flavours and colours of those. It's also natural products chemistry is what different species use to communicate between one another in positive ways, like the colours and scents of flowers that attract pollinating insects, bees, but also toxins. There's a sort of chemical warfare between a lot of organisms going on. They can't run away, some of them, microorganisms and so on, or plants. They can defend themselves by producing natural products that are toxic, that give them, can protect them from herbivores and from other organisms. We want to understand that toxicity. I'm going to give you an example of that. And we can also use that for our own benefit. For example, microorganisms, soil microorganisms produce natural products called ivermectins. And those are included in, we can now use those to protect our cattle and sheep with pour-on drenches. Plants, of course, have been the earliest medicines of humans, and we can refine those natural products, and half of our current pharmaceuticals is estimated are closely derived or inspired by natural products chemistry. And to recognise the importance of this, in fact, the 2015 Nobel Prize for Medicine was awarded to 3 natural products chemists for their work on the ivermectins and on the antimalarial compound artemisinin, which is saving millions of lives from parasitic diseases across the tropics. So it was that sort of role of natural products chemistry that I was introduced to at the University of Bristol, and in particular by Professor Mark Whiting, who was giving undergraduate lectures there. And that got me inspired to follow through on that sort of work. Professor Whiting was important in my career. I did an honours project with him. He got me that job in the flavour research industry, working with companion animals for flavours. And then when I decided that I did want to do some more formal research training, to do a PhD, he had an inquiry from Professor Ted Corbett, who was the head of this department, chemistry department here at Otago at the time, looking for a student. And what Ted Corbett wanted was somebody to work on the chemistry, some unique chemistry of New Zealand trees. And of course, being English, I hadn't come across New Zealand trees. But when I started looking into that, it turns out that natural products chemistry of New Zealand's native trees has been very important for the economic development of New Zealand. So not just their timber, but in particular from Kerry, it's Kerry Gum. So this is it's truly a resin that oozes out of damaged Kerry trees and has accumulated in the swamps in Northland. And it's stated that the early Auckland was built on gum. From 1850 to 1950, exports totaled 450,000 tonnes. and that was being used overseas as very high quality varnish. Okay. And that was found in the swamps from Old Kerry, or where they died and been growing for thousands of years. But that became mined out, of course, and then to keep that going, people started climbing the trees. And as an alpine ice climber, I was fascinated to see this picture from this book of a gum climber. So they've got picks in each hand, spikes on the front of the boots, and that's exactly the sort of technique that we were using with my friends when we were climbing the Alps and in the Darrans. But except that had only been invented in the 1960s, so New Zealand climbing could have made an even greater leap forward if we'd adapted that, if that had been taken up for ice climbing earlier on. So the chemistry, this is mainly a compound called agathic acid, named after the genus of Carry, Agathis. And it was described first from that. And I've got to introduce you to this sort of diagram, which shows the structures of natural products and all organic compounds, which are based on carbon and hydrogen, with varying amounts of oxygen, nitrogen, and then other elements. Some people refer to these as chicken wire diagrams, and you'll see why later on. But the point here is that we're carbon-based life forms, so to save time, we just draw these diagrams where we assume that every vertex, every angle on this diagram is a carbon atom, and it's carrying its requisite number of hydrogens. So it's 4-coordinate carbon, so if it's bonded to 2 carbons, it's also bonded to 2 hydrogens. So that's some of the basis of these structures. The project that Ted Corbett wanted to follow up on was from another New Zealand native tree, another endemic tree, only found in New Zealand, the rimu. Most of you will be familiar with rimu, I hope, one of our great forest trees. That grows extensively, well, it's the most widespread of our forest trees, grows all the way from Northland down to the north of Stewart Island. And the foliage of that contains this molecule, which is the one shown on the end of the Science One building, and it's called laurinine. Now that's called laurinine after Dennis Laurin, who who's actually working for Plant and Food Research now, though he's semi-retired, and he did a fantastic amount of work in his PhD study on that, determining this chemical structure. And this weird thing about this chemical structure is that you've got a central carbon atom bonded to 4 different carbons, so that's normal enough, but those are also fused together in this series of rings. And that sort of structure had been suggested as a possibility, theoretically, by Roald Hoffmann, Nobel Prize winner, one of the great chemists who he actually spoke here in 2013. And he and co-authors there had suggested it might be possible to get what they called fenestrine type structures, in which there was this central carbon atom fused in these 4 rings, and what you could have there was then possibly enough strain to flatten out the carbon. Because carbon, one of its properties that makes it really the building block of our molecules of life, is that it has this 4 bonding capacity, and it's also 3-dimensional, so this is a tetrahedral type structure. So if you're trying to squeeze that into rings and get it towards a flat structure, that will introduce a lot of strain. So they thought, theoretically, could those be produced? And so fenestranes after the window pane type structure, or a rosettane. And laurinine, once the structure of that was determined, turned out to be the only naturally occurring fenestrane or rosettane, and that's still true today. So that is the intrinsic interest. It's a New Zealand molecule, it's a Dunedin molecule. In fact, this is the crystal structure here, so that managed to get a brominated derivative and make a crystal that diffracted X-rays and determined the actual structure of the molecule, and you might be able to see there that that's the central carbon there, and it's not in fact flat. But that structure did show that there is a lot of strain in that molecule, which I'll come back to in a little. So that was the thing that Professor Corbett had set up as a research project. And because when they were trying to get more laureline to study its chemistry, they went back to another Rimu tree, and they found almost no detectable laureline in that particular individual tree. Went to another one, found some. So there was some natural variation in there. And the idea was to study that, and we might understand then what factors led to that variation. Perhaps we could understand from co-occurring compounds how you could get such a strange molecule in nature and whether it has any role in the survival of, ecological role in the survival of the tree. So my first job when I got here, actually Professor Corbett handed me on, perhaps when he first met me, I don't know, to Rex Weavers, who was to be my PhD supervisor, who was a great supervisor for that, and very encouraging, very supportive. And with Rex, we planned a campaign of sampling. But first of all, we needed to be able to measure the concentration of laureline in Rimu foliage. It's a colourless, odourless molecule, reasonably low concentration. So I had to use some of the techniques I'd learned in the flavour research and extraction techniques so that I could take lots of samples of foliage with different treatments and measure the concentrations of laureline in that. So this graph here, you can see the old sort of approach in the typesetting from my thesis, 40 Rimu trees, and analysed those by gas chromatography, measured the relative amounts of laureline, and there were some trees there which have mostly laureline, ones here which had almost no detectable laureline. And then there was an intermediate group, but these had another compound, which is actually called rimuine, named after the tree, of course, and characteristic of that tree, and found in New Zealand also, as the dominant compound in that class. And then there was some with intermediate composition, roughly equal composition of those. So, yep, there was the variation. So what was causing that variation? I tried all sorts of different factors. We looked at the gender of the trees. Rimu has separate male and female trees, male trees with the spore-releasing or pollen-releasing cones, female trees with the fruiting cones, but that didn't make any difference to the proportion of laureline. Juvenile foliage, the young trees have this attractive drooping type foliage, and then it gets a bit more smaller leaves and a different form when it's adult, but that didn't make a difference. Was it something to do with the position on the tree? So I went at Rex Weavers and I went out to the Otago coast forest just south of Tarry Mouth and got to look for a tree to climb, for me to climb. I think I did take my ice axes and crampons, but luckily I found a tree which had these two trunks, so that was easy. Could bridge up across those, climb up that, get samples up and down the tree, go up to however high felt safe, and which was quite high then, not now, and took those samples back to the lab, and I extracted those, started analysing them, and I started to see a pattern in the composition there. I could see two groups of samples with different compositions. I thought, at last I've got something that's different here. But then we went back to Otago coast forest, looked at the tree that I had climbed with its twin trunks. It turned out it was actually two trees. It was two saplings had started growing together, and they'd intertwined and grown up, which was why it was easy to climb, but it also meant there were two individuals there. We obviously had exactly the same environments. So it wasn't an environmental effect. It wasn't a height effect. It was something with genetic control. And in a sense, that's well, but that did vary the proportion around the country. So you can see for the Otago coast forest, about half of the trees had almost no detectable rimuine. Only a small proportion of them, about 10% of them, had no detectable laureline, and only a small proportion of them had dominant laureline. And it was similar from Puriora just north of Totoro. And, but over on the west coast at Hokitika, it was quite a different picture, significantly different in terms of statistics. We analysed enough trees, collected enough trees, foliage from enough trees, and there, 3/4 of those trees that we sampled were dominated by laurinine. And only one really out of 10 had low laurinine in it. I had to leave that project then. I worked on other podocarps, found other interesting systems, but I wasn't able to solve the ultimate question, why laurinine? Rex Weavers, with various students, kept working on looking for laurinine and working on the native gymnosperms and looking at related compounds. And then Don Clark, working with Rex, collected Totoro from around the country, Podocarpus, a separate genus. And he did find, using the same sort of techniques that I'd set up in the first place, I suppose, extraction, gas chromatographic analysis, he did find a trace of laurinine in a few individual Totoro trees from the west coast. And he could confirm that by isolating the compound. And he also isolated this compound co-occurring with laurinine. And that is a compound, well, they named it after the Waiho River over on the west coast. They called that Waihoense. And if you look at that structure there, these two rings here are preserved from the laurinine structure, but there's been a movement around the molecule here. That group has shifted across there. This bond has shifted to there in Waihoense, and this one has shifted to there. And this is another peculiar, very valuable property of carbon containing molecules, so they can undergo these rearrangements. And in this case, what had happened during the synthesis of laurinine, by whatever route, which we still don't understand, there had been a bit of slack, perhaps, in the biosynthetic pathway, and there'd been a further rearrangement to release some of the strain of that molecule. But we still don't know how particularly that's produced or why. It's not biologically active. We couldn't find any biological activity for that molecule. But the trees, well, we would think, in terms of evolution, they're devoting this energy, this highly specialized synthetic mechanism for some reason, but we haven't been able to work out why. So it remains a sort of academic question, but some of the techniques I'll come back to because I have applied them to a much more practical application in our current work on Manuka. So I'd finished my PhD then in 1984, as you heard, and I was very lucky that John Blunt and Maureen Munro at the University of Otago were just setting up a marine natural products research group. They'd got some really good funding, and they were looking for anti-tumour and antiviral compounds from marine natural products. So this is where we go diving under the seas now. They'd set up a group with chemists, postdoctoral chemists, I was one of those, with Rob Lake, with a cell biologist doing antiviral assays and cytotoxicity assays, with a marine biologist with diving experience. And so we were going to collect marine organisms from all around New Zealand and test those for their biological activities that could potentially develop into pharmaceuticals. I took the opportunity of learning scuba diving, so I got to go on some fantastic trips diving all around New Zealand. Fiordland was a highlight, and also down to the Antarctic ice on one trip. And that's there. I mean, you look at the ice frozen on the Ross Sea, and only 30 metres down, there's incredibly rich life on the seafloor underneath, including sponges like this green Latranculia species there. And I'd actually worked on that in New Zealand and worked on the Antarctic collection and found that being able to isolate, and I was one of the first person to report in the chemical literature, this structure here, which you can see has not only got the carbon, it's got sulfur, it's got bromine, it's got nitrogen, a really amazing molecule, I think. So collecting there under the ice was a fantastic opportunity. You have to think about, that was for the chemistry, but the physics comes into play, of course, in Antarctica. You're diving in, obviously, this is me at the crack in the ice. I think I was about to go down, but what you have to do there is you have to watch out for the physics. You've got your scuba tank on your back, the compressed air has to be decompressed and down to atmospheric pressure. And that decompression takes heat out of its environment, so you can get ice building up on your regulator there. This can have two effects. It can either jam open, which isn't too good because your air's just going out very fast, which is why we always dived on tethers or on ropes, so that we could be sure that we could get back or be got back to our entry point, our hole or crack in the ice. Or your regulator might jam shut, which is also not good when you're down at 30 metres. But we did have a spare regulator on the side for that, so I didn't have to do this, but you'd have then been searching for that to put in your mouth to get back to the hole. I found it quite nerve-wracking, actually. So there's also great diving out in the Otago harbour, and we did collection trips, came down from Canterbury, based ourselves in the University of Otago, Portobello Marine Lab. It was the old lab at the time when I started, which was, now it's a fantastic facility there, being rebuilt again though, of course. And we were diving in the Otago harbour, and we know we've got great life that we see on the surface, the seals, the albatrosses, the shags, the penguins, and they're of course all dependent on the abundant life in the seawater. And this flow of tidal flow in and out of there, and the nutrients coming down off the land around, gives fantastic growth in there. I was diving at one part of the trip between Quarantine Island and over towards Port Chalmers, and the sponges and the marine encrusting life was about this thick on those walls. You wouldn't suspect that, but just incredibly rich life. One of the places we dived was out at Harrington Point, out towards the albatross colony, and we found this fairly unprepossessing looking yellowish green sponge. But when we got this back with our other collections back to the lab in Canterbury, did extracts of those, and that showed up as having strong antiviral activity. So it inhibited the growth of poliovirus in a cell, a host cell. So that made it a top priority to study that. But you've got a sponge. It's the most primitive form of multicellular life. And so you can't really divide it into leaves and twigs. We just blended that with organic solvents. So you got this mush. Filter off the solids that aren't soluble, and you've got a solution that's got everything from the sea salt, the proteins, the DNA, all the way up to the lipids that make up the cell membrane. So it's a real chemical haystack, and we're trying to find the bioactive needle, the particular molecule that's giving the antiviral activity. So what we do for that is biological activity directed chromatography. So chromatography, we're starting to separate out the chemical components into fractions, and then we test each of those for the biological activity. And in this case, from this sponge, we only had to go through 2 or 3 stages of fractionation, and I had something that looked like a pure compound by the tests that I could do. I had about 2 milligrams, 2/1000ths of a gram of that, and it's a pale yellow oil. You could barely see that amount. I could get enough results on that at the time to show me that it was a compound that wasn't known in the chemical literature at all, hadn't been reported before, and that it was an amide, so I gave it this working name, mycalamide A, because there were some related compounds. But 2/1000ths of a gram, well, then the techniques weren't quite up to solving that structure, so we had to go back and collect again. And the first time we went back and we went out Harrington Point, it was a bit of a shock because we couldn't find any of this sponge, which had been reasonably abundant there before. And we thought, well, we haven't wiped it out. Has there been somebody's dragged the net across it, or what's happened? But it turned out the sponge is seasonal. There's enough nutrients there, and it grows fast, and it came up in spring. So we got back to diving there in spring and got enough sponge that I could purify about 20 milligrams. And then at that stage, I had enough to solve the structure, as it turned out. The first thing you do, you're starting with an unknown molecule. You need to know how many atoms are in there for a start. What's the size of the chemical puzzle that you've got to put together? So we use a technique called mass spectrometry, and that's the most sensitive analytical technique we've got. You ionize the molecules, accelerate them through an electric and magnetic fields, and very accurately measure their molecular mass. And you measure it so accurately, you can work out how many carbon atoms are there, as you see, 24. How many hydrogen atoms, how many nitrogens, oxygens, any other elements. So that was the size of the puzzle that I had to put together. And the next stage tool for doing that is a technique called nuclear magnetic resonance spectroscopy. And in that we use superconducting magnets cooled with liquid helium, put the sample in that, and once you've got that extremely high magnetic field, we can get separate signals for all the carbon atoms and for all the hydrogen atoms in the molecule. And we can understand from where those signals occur and the shape of the signals, how they all connect together. So then I could start putting together pieces of this molecule. And I got this sort of circled portion of the molecule, and I thought, well, that was quite a good piece that I could start searching the chemical literature. I knew that the whole molecule was new, but it seemed it didn't seem likely really that I'd got something entirely new. It's always possible. It could have been like laudanum. But I did a search anyway, and did that fairly late at night at the University of Canterbury. And I was sitting there, and I got a hit from that search. And that was sort of disappointing in a way. It meant there was another molecule that was similar, but it gave me the answer, accelerated the progress towards the answer. And the hit that I got was a compound that was called pederine. I hadn't heard of that. But when I looked up the chemical, where it was reported, it came from something called a blister beetle in the genus Pederus. I hadn't heard of those either. But in some tropical countries, also in Iraq and even in Australia, there are some of these beetles that release this compound pederine that has a blistering effect on your skin and can be quite a pest. And I actually demonstrated later on in the project, unfortunately, I shouldn't really be telling you this, that I was a sufficiently clumsy chemist. I hope you can just see across here some blisters across the back of a hand, which healed very nicely. That was also one of the properties of pederine that had been noted. So I knew had a closely related compound, and then I was able to put together the rest of the structure of micalamide A. And the rest of the structure was quite similar to the pederine from the blister beetle. So that's a real mystery. How can you have something in a marine sponge in Otago harbour that is similar to beetles, terrestrial beetles, in completely different countries around the rest of the world? And then another aspect of this puzzle question came up at the same time. We were working with Japanese chemists, collaborating with Japanese colleagues, and they had a sponge from the northern Pacific near Japan. And they had isolated a biologically active compound. They were working on the structure, putting it together the same way. And since we were collaborating, we shared the structures before we published them. And they had got this compound they called onamide A from this sponge, quite a different sponge. And it had the same portion there as I'd found in micalamide A, and very similar to pederine. So this was even more of a puzzle, different sponges and then terrestrial beetle. At the time, when we published that back in 1988, we speculated that it could perhaps be due to symbiotic microorganisms, and their sponges are full of symbiotic microorganisms. Not surprisingly, they feed on them basically, and some of them survive and grow within them. So we speculate on that, but we didn't have the tools of molecular biology at the time to study that. But it did turn out that nearly 20 years later, Jürgen Peel, working with John Blunt and Marion Monroe and others, did find that the pederine-type pathway, they could trace that to an uncultivated and uncultivatable bacterium in there, or a bacterial pathway. So we'd been right about that. So that was sort of a highlight of my work at the University of Canterbury. Unfortunately, that compound didn't get developed as a medicinal compound because the antiviral mode of action was by protein synthesis inhibition in eukaryotic cells. And of course, we're made up of eukaryotic cells, so we can't really afford to stop that going on. But there have been other compounds from colleagues, Peter Northcote and Chris Batsil at the Victoria University of Wellington, have found different compounds that do have potential anti-tumour activity from the same Mycale species, but different bacterial symbionts. So moving on to the most recent stage of my career, coming back here, got the job here, a joint appointment. It was an initiative between Professor Brian Robinson in the chemistry department and between Dr. Conway Powell in math technology, as it was at the time, out at Invermay. They want to set up this joint unit, which is called, at the time was called the Pint Extracts Research Unit, as you heard, Peru, which is sometimes a confusing acronym when people say they're going to visit Peru, just do a short trip. We've been working on the applied aspects of New Zealand natural products chemistry. Obviously, the marine natural products chemistry was looking for applied applications in terms of pharmaceuticals. We've worked on things that are closer to commercial development. So as you heard, we've been working with Ngāi on Taramia, on the speargrass that gives so much of a pain to some trampers. We're working on developing the perfume of that's been traditionally used by Maori into a modern product with a sustainable production system. Been working on Horopito. Most of you will recognise that bush pepper up in the town belt and so on. It's got that very pungent peppery taste, and we've looked at how the chemistry of the pungent compound and that red coloration that develops gives signals to herbivorous insects in an ecological project that we got Marsden funding for. We're working on apple diseases, as I think Richard mentioned, analytical techniques for detecting diseases of apples before the symptoms arise, so that they could be save money by not growing them on, not planting them out. One of the main research themes that we've had on the native plants has been on Manuka. Again, well, it's more widespread than Rimu. It's our most widespread native plant, growing all the way from Northland, all across Stewart Island, on the other offshore islands. And this has unique chemistry as well. Most of you will know about, I think you all will know about Manuka honey, but you'll know less about the Manuka oil. This is from the foliage on the East Cape. There's a commercial production of a volatile oil from the leaves of Manuka, and that contains an antimicrobial component. This is shown here, which is called leptospermone. That's named after the leptospermum genus that Manuka is in, and was first found in Manuka and also from some Australian leptospermum species. And that has antimicrobial activity against gram-positive bacteria. And in fact, it's got antimicrobial activity against strains of antibiotic-resistant bacteria, and we've done work on that with Phil Bremer and with Greg Cook, and that has some potential for treating sort of school sores, which are becoming resistant to mupirocin and other antibiotics. We've also looked at, and the point here is we were using that sort of technique that I had, some of those techniques I developed in my PhD work, or the concept that just because it's one botanical species doesn't mean it has to have identical chemistry all the way through its range. And that's very true, of the triketones. You see up here on the East Cape, the red dot shows that the distilled oils have greater than 15% of the antimicrobial leptospermone and related compounds, whereas just down the road in Gisborne, much lower concentrations, and around here in Dunedin, hardly detectable, much lower antibiotic activity. But these other areas have some interesting chemistry as well, distinct chemistry. For example, from the Golden Bay, we've found there's I believe it's a really attractive scented oil or flavoured oil as well, which has a spicy citrus type aroma and flavour, and it makes a delicious marinade on green-lipped mussels there, for one example. And we've also found compounds related to the leptospermone. We just reported those fairly recently. that have never been reported from anywhere else in the world. And they have, the nearest structures to those are in hops. Now, the hops that we use to flavour beer. So what we're trying to do now is look at whether, and we have some indications that these nitroketones are going to have bitter flavours as well. So what we're looking at now is finding plants, working with Te Tau Ihu, which is a group of Maori iwi across Nelson Marlborough. And we've been out collecting individual plants there, and we're just in the process of analysing those for high levels of these nitroketones, plus the attractive aroma compounds. So what we might be able to do is develop with the Maori landowners and New Zealand food and beverage companies a Manuka bitters type, a genuine sort of unique New Zealand beverage, not necessarily alcoholic, that would then have the novelty value that beverage markets are seeking. I mentioned Manuka honey that we're all familiar with. This again contains not a unique New Zealand natural product, but a very simple one, which is this 3-carbon dihydroxy, 2 hydroxyl groups, acetone, you might recognise there, dihydroxyacetone in the nectar at unusually high levels, much higher concentrations than have ever been reported in any other nectars of other plants from around the world, except for leptospermum in Australia. So, of course, they have biologically active honeys as well. And this is converted to an antimicrobial component when the honey is stored, and that gives the value, and that's what has seen this boom in Manuka honey sales, 233 million in 2015, up to 280 million in 2016 in last year. But there are various, and this is export, okay, so it's a valuable industry. But as you've been hearing on the news the last few days, myrtle rust is a major threat to that. So obviously Plant and Food and MPI and DOC and so on, we're all sort of trying to work out what we can do. My hope is that for variability of what is called Manuka across New Zealand, there will be resistant strains across New Zealand. But there is another threat to New Zealand honey in general. It's been known for a long time that since European bees were introduced into New Zealand in 1839, as early as 1857, there are reports of deaths from people eating honey that they'd found collect in the bush, or even from beehives that they were looking after. And a colleague of ours in Plant and Food Research, Mark Goodwin, has written a short book about it. And that boom in Manuka honey has led to, there has been a sort of a Food Safety Authority regulations, I'll come back to that for a long time, but the boom in Manuka honey has led to less experienced beekeepers, and one of those was actually poisoned, and various people who had some of his honey in the Coromandel area in 2008, and rather unpleasant symptoms, delirium, seizures, variable memory loss, and as I've said, there have been, not in this case or not recently, there have been deaths from this poisoning. So the question, of course, was why? Why do these New Zealand honeys have these neurotoxins in them? The actual symptoms were a clue to that, because the native plant tutu, which is various Coriaria species, native to New Zealand, had been known for a long time, is well known to be toxic. Maori knew all about the toxicity, but despite that, they managed to make a safe beverage out of carefully expressing the juice without getting the seeds included in that from tutu. But generally, there's a lot of neurotoxicity. Basically, don't try this at home. And that has the same symptoms of these seizures and delirium and so on. So they tested, and this was tracked down to a compound that was named Tooting as a neurotoxin, Tooting, from tutu. And its structure was determined, and it has been found in 1 or 2 other plants overseas, but not a large number. And it was found in toxic honeys. Okay. So then the question was, how do the bees get the neurotoxin from the plant into the honey. Of course, the obvious thing would be if it was in the nectar, but it's not. They don't produce nectar. It's not in the pollen. So how are they doing that? And there was a really good piece of field work, scientific detective work, that the researchers published in here that there were these leaf hoppers that, or vine hoppers, that actually sap feeding, and they plumb into the sap of the plant, of the tutu plant. They get the sugars that they want, and they excrete what they don't need. And of course, they're getting a massive amount of sugar. And it turns out, and this is called honeydew as well, like the beech honeydew, and that contains the tooting, and bees will collect that, and that gets into the hives. So then the food safety authorities had to bring in the regulations. So anywhere where there is tutu, which is most places where there's bush around New Zealand, and these vine hoppers, which is most of the North Island and north of the South Island, you have to get all your honey tested for the tooting concentration. There's a lot of testing being done to keep it safe. And there is a regulatory limit on that of, in fact, 2 parts per million at the time when we started doing this work. Tooting had been identified, but the original researchers on that also thought there could be, they saw sort of delayed onset symptoms of poisoning as well. They thought could be due to other components. And this was followed up by Food Safety Australia and New Zealand researchers Barry Fields and colleagues published a study in 2014 where they actually got ethics permission to feed healthy male volunteers, Wellington students, a safe dose, calculated safe dose of honey containing a calculated safe amount of tooting. And they took bloods, okay, over time. So it's a pharmacokinetic study like the ones that we do with drugs to check how well they're absorbed. And there was instantly, after they'd had their honey on toast, there was an increase in the toxin in there. And then it started to come down. But then there was this long, slow build-up over 24 hours, at 48 hours. There was still detectable tooting. And the researchers thought, yes, that is due to tooting being released from some conjugate form in there, and we needed to know what that was as a food safety issue. Okay. Unfortunately, my group, or our group in plant and food research, we got the contract to do that work because we had done previous work on tooting. So to find out what the bioactive molecules giving this sort of long-term potential poisoning, we needed some way of detecting those. We couldn't do biological assays, obviously. That's not ethical or safe. So we had to use an instrumental technique. And I've talked about mass spectrometry before. So that's the technique that you ionize a molecule, very sensitive, you can measure the molecular composition, but you can also feed in enough energy so that the molecule can come apart into fragments. And what we did with that was we had introducing the samples, separating by liquid chromatography, so there's a time separation of components there, and then mass spectrometry. And so what we were looking for were molecules that would give ionize and give a molecular formula, but we could put more energy into that and recognize a fragment that was actually the toxic tooting in there. So in this was a foliage extract, that's the tooting peak. It has this mass of the iron with various things added to it of 339. And then in a toxic honey sample, we found earlier peaks that also gave a 339, a tooting type fragment. And so these were the conjugates, these forms that we were looking for. And the added mass corresponded to either a glucose or similar fructose or similar molecule. And we also found one with 2 glucose or fructose or similar molecules. That was as much as we could tell from the mass spec, but we needed to know their full structures. So we did that by, again, getting back to the nuclear magnetic resonance spectroscopy, but we had to have them pure. So this was, again, another needle, in this time a sticky haystack. And Leslie Larson did fantastic work purifying the 2 conjugates there from a mass of toxic honey. And then we could run the NMR spectra. So we're getting signals from each carbon along here, and we're getting signals from all the hydrogens, and we could start to that same process of putting together the parts of the molecule. And so we've got an intact Tooting, as we expected. That's what's released in the body, in the digestion. But it's got bonded onto it a sugar-type molecule. In fact, this has two. And we can also use the NMR spectroscopy and tell what the actual structure is around the carbon atoms there. So we got that full structure, and we published that, and we passed on to, obviously we informed Ministry of Primary Industries about that. And the problem with that was that we couldn't actually get enough of that to give analytical standards for the full analyses. Despite getting some University of Otago research grant funding to try and synthesize molecules, we weren't quite successful in that. And the ratio so you got straight Tooting in the toxic honeys, but you've also got differing ratios of these sugar conjugates in there. So you can get different toxic effects if you've got different relationships of those. So Food Safety Australia and New Zealand had to lower the threshold from the testing for Tooting itself, which was what was being tested for in honey, from 2 parts million down, and they had to cut that by a third to give a calculated safe dose. So that was a real exciting outcome for us, that we could really make a difference to food safety in New Zealand and potentially for our export markets. So to wrap up, I've shown you that knowledge of natural products is essential for public health, as I've talked about, and also the new medicines that we had a go at that. And you can get a sort of molecular view of nature. You can link the molecular biology, the botany, the ecology together. And so I think it's a fascinating topic. And there are still other unique molecules to be found out there. Oliver Watkins, who's working with Kurt Krauss and Miriam Sharp and I, has actually shown that we haven't quite got the full structure, but that's the New Zealand glowworm that we all know about. But the chemistry has not been known. And Oliver has shown that it's a unique molecule in there as well, still to be found. So, and finally, it's me to thank all my colleagues over the years and students I'm working with now, especially my current team here. Thank my family, my parents Vera and Brian, and my brother and sisters. Thank our children and Tracy. Thank you.
I'm Lyle Hanton. I'm the head of the chemistry department. It's my pleasure to thank Nigel for his excellent talk and to make a presentation from the university first of all. So I'll do that now, Nigel. So get from the university for your lecture.
Thank you. Thank you.
It's just my job really to sum up. And Nigel put a date on things, but I well remember actually the day Nigel got his PhD, because we went round to Rex Reaver's home and we played billiards on his table. So I remember the day that Nigel, it makes me very old. I remember the day that Nigel received his PhD, and thank you for your lecture tonight, Nigel. It was great. I think to have some of that history of the laureline molecule, which we proudly have attached to the end of our building now, fleshed out. And I think you can see the interesting history, still yet to be resolved, the actual use of laureline in plants. But there it is on the end of that building there. You know why? Because it acknowledges, the reason that we chose laureline was to acknowledge the strong history that the department has had in natural products chemistry. And Nigel is one of the many members of a chemistry team who have pursued those sorts of products over the years and who continue, particularly I think with the award of the Nobel Prize in 2015, we expect a resurgence perhaps in natural products chemistry. So Nigel, thank you very much for your lecture. I think it's really interesting to see the impact of the work that people do. And for us in the chemistry department to have a CRI embedded in our department through the Peru unit, the Plant and Extracts unit, now has been in the department for 20 years. Nigel and his staff support interest in natural product chemistry. They provide supervision for students and are engaged in the teaching and just the general life of the department. And it's something that we've, I think, with your leadership in that role, have engaged really seriously with the department. And I think it's a very synergic relationship. So thank you.
Symbiotic.
Symbiotic, if you like. And so it just remains now for me to invite you all to the staff club, where you'll find the things that Nigel talked about, the means to celebrate this lecture, I think I would say. Once again, please thank Nigel for his excellent lecture.
Thanks, Brian.